Hello, I'm Mike Edwardson. I'm Professor of Molecular Pharmacology at the University of Cambridge. Today I'm talking with Professor Alan Cuthbert. Alan is distinguished for his work on epithelial ion transport. He has a particular interest in cystic fibrosis, and he's published over 300 research papers. He's a Fellow of the Royal Society, and in 2005 he was awarded the Wellcome Gold Medal by the British Pharmacological Society. He was Sheild Professor of Pharmacology for 20 years. He was also Master of Fitzwilliam College for 9 years. So Alan, if we could begin at the beginning, I think I'm right in saying that you were the first member of your family to go to university. What was it that first switched you on to science and to biological science in particular?
Well, that is true. I was the first person in my family. I think higher education was not something they ever were able to contemplate. And as for what switched me on to science, I was fortunate enough to get a place at a boys' grammar school, and I just thought science was wonderful. But science meant chemistry, physics and mathematics. No biology at a boys' school. That was all rather sissy in those days. We are talking about quite a long time ago. And so I had very little interest in biology. I really wanted to be a chemist. And I thought being a chemist, you could actually make new substances of matter that never existed before. And this was an exciting and thrilling thing to do. Why you'd want to make them, I hadn't really considered at that stage, but there we are. That's what I really wanted to do. It turned out that possibly the thing I could do was become a pharmacist. Mm-hmm. So I went off to the local tech, actually. Eventually became a pharmacist. Mm-hmm. And then I realised that was a commodity you could do things with. And so, but when I was doing the pharmacy course, there was a teacher called Bob Finney, and he said to me, you ought to think about biology. Biology is going to get very important. And I was talking now, 1951, 52. It was a prescient time. I mean, people were on the edge of DNA and all sorts of things. And so I thought about that, and I ended up doing chemistry and pharmacology.
So your studies were interrupted by a 3-year stint in the Royal Navy, where you were a lieutenant. So how did that affect you in the long term?
Well, I knew I had to do it. I mean, in those days one got deferment if you were going on to higher education until such time as that finished. And when my time came, as it were, I'd already met Hetty, my future wife. We wanted to get married and all sorts of things. I didn't have a bean, and neither did she. And I thought, if I tried for a short service commission, that might be a good way to go. I got a commission in the Royal Navy and spent 3 years in the Royal Navy. It wasn't that exciting, but I didn't get much sea time in. Not that I actually wanted much sea time. But,
So after that you went to the School of Pharmacy to do a PhD under the, I guess, notional supervision of Gladwin Buttle.
Yes.
So why don't you share some memories with us of that time? There were some quite colourful characters and important characters at the square at that time.
Yes. Well, one of the hardest jumps, really, is coming out of the forces and then convincing somebody you really want to do serious science. And somebody said to me, all you want to do is you want to write to all the top people you can think of and go and see them. And this is what I did. And I did have some ideas about what I wanted to do in research. I went to see a Professor Yoffie in Bristol, who had advertised some interesting job, which I thought I could probably be involved in. And I went to see Sir Charles Harrington, who was head of the Mill Hill, the Medical Research Council's institute. And he didn't think much of him either. And I wrote to Henry Dale. I mean, Henry Dale was already retired, and he must have been 75 or so at the time. It was extraordinary. He gave me half a day. I mean, you know, and he listened to what I wanted to do, and I think maybe he thought it was a bit unusual, this chap out of the Navy wanted to talk about things and having some ideas about what he wanted to do. He told me he could almost certainly get me a nice post at the Wellcome Foundation. And I said, well, it was very kind of him, but I didn't really feel that I wanted to be in industry. And he did remind me he'd done quite well himself for being in industry, because that's where he was when he won his Nobel Prize. But nevertheless, he says, well, you need to see my friend William Feldberg. And so he sent me off to see Feldberg. And of course, I should explain, I was going to be out of the Navy in less than 2 weeks, and I wanted the job to go to. Feldberg was really moved all he could. He phoned various people and so on, but nobody, these fellowships were closed and the dates had passed and all sorts of things. So he said, well, the best thing is you better go and see my friend Buttle.
Right.
So that's how I got to Buttle. Now, Buttle was a great eccentric. He had been part of Montgomery's team in the desert during the war. And as I guess a sort of a reward, he was Professor of Pharmacology at the Square in London, Bloomsbury Square, and then Brunswick Square. And it was a great department of pharmacology at that time, one of the best. Mm-hmm. Buttle was a friend of Harold Reinhardt, who had been a tank commander in Rommel's army in the desert. And I often wondered, and I never asked him, whether he'd actually met Reinhardt in the battle in the desert. Anyway, between them, they cooked up this Pfizer fellowship, which Buttle said I could have. And it was an extraordinary interview he gave me. It was arranged I should meet him at King's Cross at about 11:45 on a Saturday night. He took me around to the square. He had a huge bunch of keys. And he showed me a small empty room and said, would that be adequate for what I wanted to do? And I said it would. He said how much money I could have, and that's really how I started. I mean, there was all these people, Jeff West, Bill Bowman, Mike Rand, and so on.
The all became very distinguished.
Yes, indeed. I mean, Mike Rand I learnt so much from, because he had a new idea every few minutes. And then every few minutes later he would reject the idea because he'd thought more about it and so on. But occasionally he didn't reject the ideas, and I got that way of thinking about things from Mike, really.
So what were you working on?
Well, I had this idea that, to go back a bit, about receptors. You see, Butler never believed in receptors, and I'm not sure Dale did. Dale used to talk about receptive substance. And yet, in embryonic tissues, he seemed to have specific receptors in the sense that you could antagonize various things on the receptors, even get kinetic constants that convince you they were real. And often, you know, because so many receptors, or the ones then investigated, were to do with neurohumoral transmission of one sort or another. This was in tissues didn't have any nerves. I mean, and what were they there for, and so on. And it just seemed to me a fascinating thing to investigate. So that's what I was doing. And for embryonic tissues, I did most of that work just by getting fertile hen's eggs and put them in an incubator, which Gladwin had kindly provided. Managed to get a grant from the Egg Marketing Board to pay for my research. And that's how I started and completed my PhD, largely using the amniotic muscle as a check.
You moved to Cambridge, to the Department of Pharmacology in Cambridge, and at the time it still had close links, I think, with physiology here, and it was a very small department under the leadership of Arnold Burgeon. So that must have been quite an exciting time with a new department emerging out of physiology.
Well, it was, but it wasn't quite as you put it. When I was at the Square, Gladwin Butler he used to say to me, how are you getting on? I'd say fine, and he'd say fine. And that was that. But I don't think he really knew what I was doing. And Edith Bulbring, who was a very distinguished pharmacologist at Oxford, used to visit the department from time to time, and obviously Gladwin had said to her, go along and see that young chap and tell me what,
what he's doing.
So she must have reported positively, and so that was good. She invited me to go to Oxford, from time to time, because she thought I should make a sucrose gap electrode and explore the amnion muscle in the sucrose gap. And of course it was just the right thing to do. On one of my visits to Oxford, there was a visitor from Canada there for the day. It happened to be Arnold Burton. And I spent some time talking to him, and I thought, a terrifically bright guy who knew pharmacology and where he thought pharmacology should be going. And then, sometime later, he was appointed to the chair at Cambridge. He often tells me now that he cannot believe why he actually accepted that chair, because, as you've just alluded to, the department was almost non-existent. Had no space, no staff, no anything very much. Anyway, when he was appointed to Cambridge, I thought I wouldn't mind working with that chap. And I would watch out for any posts that he advertised. And quite quickly he did advertise a post, and I was fortunate enough to get it. I never did work with him. But that's how I made the move.
And how was it being in the department at the time?
Well, I mean, it was, the department had virtually no space. And Arnold said about getting space for the department. Originally, he got a grant from Henry Dale, or engineered it from the Wellcome Trust for a very large wooden hut on the lawn in the front entrance to the Downing site.
Right. This is the Dixon hut.
That's what we call the Dixon building. And that about trebled the department's space, this shed, as it were. And later on, he saw Henry Dale again and got another building, which was up at Addenbrooke's, which again was a great improvement on what we had, but of course nowhere near where we wanted to be.
So it was around about that time that your interest in epithelial ion transport began, I think. So what triggered that switch from what you were doing for your PhD to ion transport?
I tried to survey what was going on in pharmacology. It went in a strange way at that time. Everybody would be working on catecholamines, and then all of a sudden that would stop, and everybody would be working on 5-HT, and then it might be histamine, and so on. And most people followed the crowd. That was the first thing. The second thing, nearly all of them ended up recording something where tension was recorded in a piece of muscle. And I really just thought to myself that there really ought to be something else you could record. Surely other things were going on besides muscle shortening, There were already people at it. Don Jenkinson, you see, at University College, was putting microelectrodes into liver cells and finding, all sorts of things happened with adrenaline, noradrenaline, and electrical signals were happening, but of course the liver just sat there. So clearly there was a whole pharmacology wasn't being looked at, and so on. Even in the department, of course we had grown to about 4 or 5 by this time, and Keith Matthews had joined us from Canada, and at Arnold's suggestion he was putting microelectrodes into endocrine cells, and for instance getting all sorts of signals which have never been recorded before from, say, pancreatic islet cells in response to glucose. So I think I was driven by thinking there must be more to it than just recording from muscle. Everything goes in through some barrier, and everything you lose goes out through a barrier, and there must be processes that are worth looking at and are modifiable by drugs.
Yeah. I remember at that time going to your final year lectures and finding them very inspirational, and it would seem to be a very exciting time in the field. Do you want to tell us a little bit about your approach? And
it was an exciting time with the epithelial sodium channel. And there was a good drug. You know, there are not many drugs with as clean a pharmacology as amiloride was.
Yeah.
One of the things that surprises me when I read a modern paper about epithelial sodium channels, I realised I didn't know anything about subunits. Nobody knew anything about subunits. I didn't know what controlled the sodium amiloride competition in some sodium channels and not others. And yet, I was only a few weeks ago reading a modern paper because there are still people trying to produce better and better amiloride-like drugs. And in the introduction it said, of course, amiloride works by an exofacial interaction with the channel. And then it says, as first described by Cuthbert, 1976. And that's not a word I'd ever used.
No, quite.
And somebody had invented this thing, and so I read more. And yeah, I realised, I went to look at what was in the paper, and I was quite pleased with it, I suppose. It was in molecular pharmacology, and it was a single author. Mm-hmm. And I'd looked at pH effects on amiloride actions and worked out how I thought amiloride blocked the channel, and what's more, why benzamyl was 10 times at least better than amiloride. Mm-hmm. Exofacial interaction.
There you go. Okay. You became reader in pharmacology in 1973 and Sheild Professor in 1979, head of department, and you held that post for 20 years. What were the challenges that you had to face when you first took over the new department?
Well, let me say a word about readership, first of all. I thought it was the best job in Cambridge, be a reader. Because in those days, there weren't many readers. There was only about 8 or 9 a year made by the university. It was a post in which you were supposed to concentrate on research. You were not allowed to dabble in administration. And you weren't allowed to do too much teaching, and neither were you allowed to do much college work. So, of course, you weren't paid that much either, but I thought it was really a super job, and I enjoyed that time very, very much. So when I took over the department, I think I had 3 main objectives to achieve. First of all, to get the department properly accommodated. Secondly, to make the department financially secure. Now, I should explain that. I mean, what I mean by that is to be opportunistic. That is, if a good, terrific postdoc appeared and there was no money, there would be money to support such a person. Or if there was a piece of equipment that really would forward the research of a whole group of people within the department, then you could just get it without waiting for a round of this or somebody writing a research grant. And the third objective really follows from the first 2, and that is to increase the academic profile of the department as a whole. Your
major legacy in Cambridge really is what we still call the new department, the new building down on Tennis Court Road, which you're responsible for getting built, basically. What were the problems that you encountered there in getting the funding and getting permission to put it up?
The difficulty was the university was not sympathetic. In fact, I don't think the university has ever been particularly sympathetic to pharmacology. And we ended up with a research facility 3 miles from the centre of town, which was a long way from where students wanted to go, or which were close to the departments with which we could collaborate and cooperate. And the trick was how to get back to the central part of the university where the other biosciences were. And how do we do that? One of the key players was Lord Butterfield, at that time was Chancellor, and he was telling me that some organisation, something to do with cancer research, I don't know if it was Cancer Research UK or whatever it was in those days, were looking for a small facility on the Addenbrooke site to do something or other. And so the proposition was, could we sell the existing research facility and take the check, as it were, and put up another building somewhere else. It wasn't enough, of course, and I had to take a loan from the university, or the department took a loan of something over £1 million to actually get the building put up. That led to a problem because the Department of Architecture thought it was not a building of great architectural merit. One of the most frightening things I've had to ever do was appear in the Senate House to defend this building. And I still would defend the building. I don't think you should build laboratories for perpetuity. 30, 40, 50 years down the line, they're not fit for purpose because the purposes have changed. Sure. Those departments on the Downing site, for instance, that are in listed buildings have the most terrible trouble. Not what you want in modern science buildings. Anyway, we got the new building and, as you mentioned earlier, it still seems to be called the new building.
Part of the new building was occupied by a research group from Glaxo run by Pat Humphrey. How did that cohabitation work?
Well, I think it worked quite well. It was always my intention when we put up the new building to try and have a academic industrial interface, both for financial and for scholastic reasons. And I think it did work on both those fronts. Financially, of course, large amounts of money, which the department wouldn't have had, became available. This allowed the department to pay off its loan that it had taken from the university to get the building up in the first place. Secondly, it also gave one the freedom of, if a brilliant young person appeared that you wished to take on, you could do so, and in the hope that their posts would eventually become available. The contract was for 10 years, and during that period there were a number of collaborations between the two groups, and of course the lease ran out just after I'd ceased to be head of the department, because if I'd still been there I'd have tried to renew it, not necessarily with that company, but perhaps one with whom we had more common ground than we did with them. Another result of having them there was we were able to put another floor on top of the building, so that all in all I think it was very positive for the department.
So in the late 1980s you turned your attention to cystic fibrosis, on which you still work. Mm-hmm. What was it that first interested you in CF, and what approach did you take?
Well, it wasn't that much of a change if you think about it. First of all, I mean, what you're suggesting is I changed from looking at sodium channels to looking at chloride channels, which I guess is to some extent what I did. On the other hand, I'd been working with chloride channels in the kinin area for some time with Harry Margolius and his group. But as you well know, in cystic fibrosis, one of the first ways you can detect somebody has that disease is that mothers always complain that when they kiss their young babies, they taste salty. And that was one of the diagnostic features before we knew anything about the genetics of the disease. Now, the idea was that the sweat was secreted in the secretory coil and it couldn't be reabsorbed because the sodium couldn't be reabsorbed. And there was talk of circulating amiloride-like substances. So this was spot on what I'd spent a number of years being interested in, the amiloride-sensitive sodium channel. And it wasn't until 1983 that Quilliam showed that it was the chloride channels deficient in CF. And of course, if you can't absorb the chloride, then the sodium can't follow, so it still tastes salty. The other thing is we were terribly fortunate being located where we were, close to Wellcome CRC. because that's where Martin Evans was, and him and his stem cells, which he more recently got awarded a Nobel Prize. And he was able, with his technologies and his group, to produce transgenic mice with this disease. And of course that led us down that pathway, thinking that in fact gene therapy might be the way in which this disease was dealt with. Well, of course it's still not dealt with, and in my view I think gene therapy for this disease is a long, long way off. And I think a pharmacotherapeutic approach is far more likely, and indeed currently there are some really quite promising drugs in clinical trial.
So you think the prospects are good then?
I think they are better than they've ever been, and give some considerable hope to people with this disease.
Mm-hmm. So in 1991 you became Master of Fitzwilliam College, which is where we are today. So at the same time you were running a college department and a lab. So how did you do that?
Well, I won't deny it was a very busy time, but a very enjoyable one. Living in the college, of course, in the lodge, was a great help, because it meant that you were on the spot for many of the things you had to do. But I was able to cope with all this, really, with the help from a lot of people. In the lab, for instance, I usually got in the lab most days and did something, but when I couldn't, there were sort of semi-permanent members of my team, like Leslie McVinish and Margaret Hickman, who kept things ticking over while I was not able to be there. And similarly, in college, there were I had an outstanding bursar, and I had a series of excellent senior tutors. And you know, if a master's got a bursar and a senior tutor that are working in unison with him, then I think that's half the battle. And of course, my wife was an enormous help while we were here, because she busied herself in many, many ways around the college, and sometimes was able to indicate to me things that if she hadn't been doing that I might have missed. So it all worked really quite well, but it was busy, but extremely enjoyable.
Did you find that there were significant differences in the skills you needed to run a department?
Well, not really. It needed increased income, and I took several initiatives to start to raise funds for the college, which has gone on ever since. And the college also needed many new buildings. It was incomplete, as we all realised. Now, particularly I was keen to initiate the thing which eventually led to the auditorium and so on. And then we see the college as it is today, more or less complete, but still requiring support in other ways. And so I'm not sure it is different. And then the third component is to elect people to the fellowship, and to elect students or select students that are as good as you can possibly have. So I'm not sure there's that much difference between what you need to do with a university department or with a college.
Since your retirement in 1999, you've maintained an active research group in the Department of Medicine. So what is it that keeps you going, and what are your objectives now?
I really enjoy doing research, both doing it myself as well as alongside other people. What keeps me going? I don't know. I have more ideas, I think, than I've got time to get done. And 3 years ago I went off to work with Jeff Wine in Stanford. I've learned a technique which not many labs in the world can do, only a couple in the States, and now in the lab here in Cambridge. So that it's quite exciting with this new methodology. We can measure things that's not been possible to measure before. And what are my objectives? Well, I'm still pursuing a possible pharmacotherapeutic treatment for cystic fibrosis. And we have a number of compounds which are causing some interest amongst other people.
Mm-hmm. I know you're passionate about the discipline of pharmacology. And over the last few years, a number of pharmacology departments have disappeared, having been merged with other departments. And this process shows every sign of continuing. I mean, what's your view about that? I mean, does pharmacology have a future? And if so, how can it be secured?
Well, I think it's very sad, but you are absolutely right. There aren't many pharmacology departments left in the UK. They're subsumed into other bigger conglomerates. I think it's nothing about pharmacology in particular. I think it's what happens to small subjects. I think administrators and organisers of institutions find it more easy to deal with fewer people than a lot of people, and so by amalgamating things together, there are fewer people they have to deal with. Now, I think it's very destructive for disciplines such as pharmacology. It's disheartening for the people who work in those departments. It also leads to, or it's imagined to lead to economies, and sometimes does, because then you can have this awful word, you can have things like service teaching, when, you know, physiologists can say perhaps teach physiology courses and incorporate some pharmacology and all that sort of thing, and of course it takes the edge off the discipline, and there is a consequence really of I think the shortage of funds and what's been happening recently, of course, and what is going to be happening with academia in this country in the next few years is in fact very, very serious. To my mind, there's only one sort of way in which one can combat this, and that is to be, you know, to have private finance and be financially independent. And of course, you asked a question a little while ago about the academic-industrial sort of collaborations which we had in the department in my time, and that's the most surest way to remain independent, is because you can overcome the difficulties that various severe economies will be facing, all departments, but particularly small ones.
Mm-hmm. Mm-hmm. So we're now in the post-genomic era, and we were promised a big upsurge in drug development. But it hasn't really happened yet. I just wonder what you think about that, why that is, and where the new drugs are going to come from.
Well, you're quite right. I mean, a very good index of how many new drugs are coming on the market is to look what the FDA approves, and it's not very many each year now compared to what there were years ago. I think the drug industry is in a turmoil at the moment. I mean, as we hear, many people are being laid off in the big pharma companies. Pharma companies are concentrating, if they have a cardiovascular group, say in the UK, they no longer have one in the US. If they have a GI group in the US, they won't have one in the UK, and so on. So it's not really a time to be thinking of designing drugs for patients specifically based on their genetic makeup, and I'm not sure I think it's even necessary. I mean, it's just a few weeks ago that a major drug was withdrawn from the market, an oral hypoglycemic agent, of which tens of thousands of people in this country and all over the world were benefiting from. It was withdrawn because very few people, a very few number of people, had some adverse effect. And it just escalates the cost of drugs in a way that's quite outrageous. 60% of the cost of a new drug is in phase 3 clinical trial. That could be scrapped, really, and that, you know, post-marketing surveillance would be the best way of finding out or detecting the very few people that were intolerant to a drug, and this would bring down the drug prices and would make it more possible for the drug industry to produce new drugs. And I think also the drug industry's got to concentrate on drugs we need, not Me Too drugs and drugs for conditions that we already have.
Cool. So you've had a long and distinguished career. What advice would you give for somebody starting off now? What does it take to make a good biological scientist and a good pharmacologist?
No, I'm not sure that aged people like me should give advice to young people unless they particularly ask for it. People who are going to be good researchers have a feeling that that's what they're going to do and will do it. The only thing I would say to them if they asked me was really try and understand what it is you're interested in, what it is, how you want to spend your working day. Do you want to be fiddling about looking down a microscope all day, or would you want to be doing some sort of different operation, or would it be a chemical thing you want to do? Or you've got to find out what sort of person you are. And then when you've found that, find a topic within that you really want to pursue. And then find somebody that's pursuing it with the same enthusiasm that you think you have. Mm-hmm. And then try and persuade that person to take you on as a junior, a novice, a learner, and so on. Mm-hmm. I think researchers are born. I don't think they're made.
Okay. Alan, thanks very much. It's been a pleasure to talk to you.